Conducting behavioral procedures to assess impulsivity in rats provides insight into the environmental, neurophysiological, and neurochemical basis of impulsivity. That is, about the factors that trigger and underlie those behaviors.
This protocol allowed the study of impulsivity with a good balance of efficiency and accessibility, and the extraction of data suitable for quantitative analysis.
The performance in these protocols may be considered as an analog of impulsiveness in humans. Hence, human versions of them serve for the diagnosis and treatment of related psychiatric disorders, such as ADHD or pathological gambling.
Demonstrating the procedure with Patsy Moreno will be Gustavo Lago, a technician, and Pablo Saavedra, an undergraduate student from our laboratory.
After beginning the food restriction regime, add 60 food pellets to the food receptacle to habituate food neophobia before introducing the rats to the conditioning chambers without the initiation of any protocol for 30 minutes to habituate the exploring responses. After the habituation stage, introduce the rats into the conditioning chambers for 2 additional daily 30-minute sessions of delivering a food pellet every 45 seconds to help the rats identify the source of the pellets. For intertemporal choice and differential reinforcement of low rates of responding, or DRL protocols, place 1 or 2 levers into the chamber and deliver food for every lever press in conjunction with a free food pellet delivery every 45 seconds.